Hi everybody. So I wanted to make some videos for the last couple classes because it was a lot of lecture and it was a lot of, it's a ton of material. So I really wanted to make sure that we were kind of on the same page with everything. So I'll just quickly go over things here. This video will hopefully be good for you to like pause and, take notes if you want some more clarification, things like that. So the main point of this is to understand not only how cells communicate, but how signaling cascades are what really are the triggers for genes turning on. And as we get more into genetics, we'll see that genes aren't always on, they aren't always off. It takes environmental stimuli to turn a lot of them on. So how cells communicate at the local level is super easy at first. Basically, if you are connected, you're going to exchange material between two cells. You can do this in animal cells, you can do this in plant cells, and everything will work out. The other way that you directly signal something is through receptor binding. So both of these cells have genes that are expressed as proteins on their surface. Oh, I don't know what that is. And each of these genes fits like a key and a lock. And when you bind, there is going to be an effect. That's what these arrows are. This effect is going to cause some kind of change in this purple cell right here. And that's how this green cell is communicating with this purple cell to do something or to change its genes, something along that side. So this is the key here is that there is some kind of direct contact interaction, cell to cell, just like right there. And this is really B right here. This is the real direct way you're going to see a lot of things happen. Most of the stuff in this lecture is going to talk about the long distance signals, but a ton of what's happening is cells basically interacting with each other. Okay, so this is long distance, and the key here is that some kind of signaling molecule, usually a little protein, a hormone, a neurotransmitter, something, they have to be secreted outside the cell. So remember that the Golgi packages up proteins like this, and it puts them in these little vesicles. The vesicles can go, and they can interact with the cell membrane and just burst out, like we see here and here. And the big deal here is that these little molecules can go off and they can find different cells and go inside them and start triggering some kind of response or interact with a receptor on these cells. This is paracrine. This means that nearby cells are going to get this signal. B, synaptic signaling, is just an example of paracrine. It's just cell exchanging to cell with small little signals here, and in this case, neurotransmitter. It's just special because these cells are specially designed to interact with one another, and it's just a good interaction, because it's cool. This is still pretty local signaling, as you can see from up here. It's not super duper long distance. The third kind, I don't know where it went. All right, so the third kind of signaling that's in here is called autocrine signaling, and it's a version of paracrine. All that autocrine signaling means is that all these molecules that a cell secretes, they can go back in and cause this cell to have the reaction as well. So true long-distance signaling is usually going to come in the form of hormones, where these little protein signals, these ligands as we'll see them called, they're going to actually go into the bloodstream goes everywhere, and eventually they're going to find their target cell. Now only the target cell with the right receptors on its proteins, or on right protein receptors on its cell, they're the only ones that are going to uptake this signal. This signal is going to be ignored by any cell that doesn't need it. But this is the true long-distance way of getting a signal across the body. This is kind of what we talked about with adrenaline. Okay. So we're going to discuss those 4 specific types, but really, if I want you to know anything for the test, it's just knowing this basic system of how you translate a tiny signal outside the cell into an activation of some kind of physiological response. First things first, this signaling molecule is going to interact with this receptor protein on the outside area of the protein that's outside the cell. When you make this bind, the 3D shape of this protein is going to change. And specifically, it's going to change in an area down here inside the cell that interacts somehow with a relay protein. Now the relay proteins are important because they're always around. They're only waiting for this section right here to become active, which is only going to become active if a signaling molecule binds to it. Once you have your first relay protein going, it will go and turn on the next relay protein, which turns on the next relay protein, and so on and so forth. Eventually, you get into some massive cellular response. Usually you end up binding a protein that is meant for turning on a gene that physically interacts with the DNA, and it's going to actually it's going to actually bind up to some kind of DNA up here, and it's going to turn on this gene, for example. That gene will go on and have some role in the cell. So the cascades that we talked about, they're this transduction, but to initiate a cascade, you initially need to have some kind of reception, some kind of interaction between the signal and the receptor. And this kind of highlights what we talked about, is that shape changes are what turn these proteins on each time, and that's how they can become a different structure that is now active each time. So we start with G-Protein Coupled Receptors because they're easy as far as it goes with they're double-sided. That's really what I want you to know here. There's a signal binding molecule side, and there's the side on the inside that's going to interact with your first relay protein, in our case, which is a G-protein. So it's not all of these signaling receptors are always going to be G-proteins. I think it's just a good example to start with, and you will see this in physiology if you go on. So as long as you know there's two domains, one here, one out here, should be good. And they kind of end that when you bind up here, whatever your signal, this area turns on and is ready to bind with relay proteins. All right, so main thing with G-protein is that you start with the system, it's all inactive. No signal has come in. Once you get your signal, this activates the G-protein receptor. The G-protein itself shows up, and what happens is it first binds right here to the active receptor. Next thing that happens is a GTP molecule, which is kind of like ATP, shows up, kicks out GTP, and binds the now open section of this protein for GTP. The GTP activated protein, which has basically just been turned on, now goes and activates another relay enzyme. This enzyme goes on, a cascade forms, and a cell response essentially will eventually happen. Some kind of DNA will change. Once this is all over, we just return to the basic off stage that we were always at. All right, so RTKs, or receptor tyrosine kinases, they're pretty much the same thing. Signaling molecule goes in, binds some kind of receptor, and everything starts. Key here, though, is that you need double signaling molecules to come in, and these receptors on the surface of the cell, they actually bunch up together, they fuse, and they form what we call a dimer. So di is 2 in Latin. You have a dimer. What that does is activate all these amino acids right here that are tyrosines. So remember, each amino acid has its own little special shape, and there's 20 of them. Tyrosine happens to be the very best one that can get phosphorylated. So an enzyme, or a kinase in this case, shows up, and it takes ATP, and it's going to put ATP and take one of those phosphates from each of those 6 ATPs and put them on these tyrosines, because tyrosine is the best thing that can get phosphorylated. And remember that ATP and phosphate groups, they're just really easy to add, and they're really easy shape change right here for us to do. So that's what activates them. It's not necessarily an expenditure of energy more than it is like a changing of a shape that allows it to turn on. Because once you have these phosphate groups here, now the activated relay proteins, they can bind now. They can't bind, none of the relays or the cascades can start. They can't bind to any of these blank tyrosines, even if the dimer is there. So if you don't have the enzyme that's putting on ATP phosphates on here, you won't have a response. These start the relay ends with a cellular response. 3 is pretty easy. Remember, we talked about channel proteins that allow ions or other big things into the cell. If they're closed, the ions can't get in, and nothing's going to happen. If they're open, and maybe they only open in response to a signal, like this ligand right here, this little signaling molecule, then they go in, and then the cellular response is triggered by these ions. There's a lot of steps in here that are pretty ambiguous, so if you just let in a bunch of charged ions, something is going to probably change. So this one's a little simpler and good to know. Lastly is kind of the strange example of intracellular receptors. In this case, if you have a signal that's actually pretty hydrophobic and it can just cross the membrane, remember hydrophobic things can just happily cross the membrane because the membrane is hydrophobic. They find their receptor partner protein inside the cell. They immediately go on and can actually start affecting DNA really quickly. So there isn't as much of a cascade in intracellulars, at least in this example. But the key is that there's no surface protein here. It just invites itself in. And only a few hormones are that easy to get in. Like testosterone, for example, is necessary in a bunch of cells in male mammals, for example. All right, so to learn more about the cascade, we've seen the signaling and the receptor protein. Once they're turned on, you activate the relay. The easiest example of relays is just that you activate a kinase. And all kinases do is add a phosphate group from ATP. Remember, there's 3 phosphates here. You take one of them and you plop it onto number 2. Number 2 is now activated. This phosphate has changed to the shape of number 2, so it's active. It takes an ATP and it pops an ATP phosphate onto protease kinase 3. This can go on in many steps, and eventually you activate a protein that's actually going to activate something in the cell, turn on a gene, cause the release of something like that. So if you want more clarification on kinases, come see me, but hopefully it's clear that each kinase is only going to be activated once it gets the phosphorylation on it, and then only once it passes that on is the next one on. So it's basically just a big set of relays of on switches. Okay, so we talked about this, is that even a single molecule of signaling receptor is exponentially going to be relayed, and that signal is going to be amplified. So a single receptor can get bound, but that can attract 10 of these proteins. Each of those proteins can activate 10 or 100 of their secondary signaling molecules, in this case cyclic AMP. So this is a pretty specific pathway. You don't need to know it. Just look at the numbers here. The numbers of the molecules you're activating continually increase, and that's to ensure that the signal is highly amplified and that there's no going back. However, the best way to eventually stop this cycle is that if there's a lot of steps, there's a ton of opportunities for all kinds of enzymes to come in and stop each one of these steps, or inhibitors of some kind. So there's lots of good, well, that's supposed to be an inhibition. There's lots of good spots you can inhibit here. You could have an inhibitor here. You can have an inhibitor here. So we talked about feedback regulation. Glycogen itself could, for example, inhibit here, inhibit here, inhibit here, inhibit here, once there's enough glycogen released. So that's the other key, is that multiple steps means that you have multiple opportunities to stop this process once you're done. So this slide's probably the most important for the basic idea of reception, transduction, and response, because it shows that the final stage, this response stage, usually means that the cascade is going to activate what we call a transcription factor. A transcription factor is a gene that binds other genes. It's a protein that binds DNA and activates that DNA to become active. That's the key, that's usually the key response here. So this is a good slide too. We've basically been working with this model where a pathway leads to a single response, there's a single pathway. However, there's tons of things that these pathways can do. Some of the relay molecules can cause double responses. Some molecules are going to compete against each other and actively fight each other. So we see this with apoptosis. And another one is that different receptors, I should do this. Different receptors on different cells, see how this is kind of a different shape than this one that we're talking about? They're going to elicit a different response because this is going to bind to a different cascade. See how this is different than these little ones over here? And a different cascade means a different response. Okay, so we talked about apoptosis and what it is. It's a very controlled pathway, and it's when a cell that is unhealthy, let's say over here, it needs to be basically be taken out. And you can see the nice organization and the cuts here that's everything making. It's not just spraying everything everywhere. And that's the key point down here, is that you don't want cells just exploding, because these are full of acidic enzymes, chemical components that shouldn't be outside the cell because they can go damage the other cells. So this is a nice clean way to get rid of cells. And you want to get rid of cells when they're bad, definitely. Big thing that we talked about is that this can come from the outside, where a T cell or an immune cell is going to bind up receptors, and we're going to have our traditional kind of transduction, like big cascade pathway. We saw that caspases get cleaved. See, this is a pro-caspase. What that means is that when you cleave this bond right here, that's when the gene is active, is when you only have one of these little bits. When that is active, it goes and cleaves more caspases. And remember, caspases, once they're around, they start chopping all of the DNA, all of the proteins, everything, and that's what really kills off the cell. The other way is the intrinsic pathway. So this is less about what we studied, and it's more about mitosis being good. So we know p53 already. If a gene like p53 notices cell stress, it starts killing the cell by opening up pores in the membrane with genes like these. Now, you don't need to know the names of the genes. p53 is probably the only thing that I would put on the test, is that once you start causing leaks in the mitochondria and this is only going to be active if the cell has tons of stress or DNA damage. That's the only reason this would happen. So if you plug up if you open the mitochondria up, this poisonous cytochrome C comes out a few transduction steps later, and you start activating more caspases. Oh yeah, and then here's this kick-ass death complex right here. That's a pretty cool name. This starts not only activating caspases to chop the cell, it actually starts opening holes inside the cell membrane. Stuff starts leaking in, the cell dies. So this is a good example of a pathway because you can see there's not really a good way to stop it if it's really committed to going. So this is a good example of how this pathway is always ready to go, this death signal. But if there's no signal binding the receptor, everything's going to be fine. Everything's going to be inactive. We're not going to chop anything. If you get the death signal from some molecule, usually a T cell, usually an immune cell is going to tell you when to die, basically. That's when we're going to start activating everything. So SED is another name for caspases, but essentially you have the same thing, a cascade. I don't know why that shape ever happens. You have this cascade that happens. In this case, it's a cascade. It's not a kinase cascade. It's more of one that is going to chop and activate genes that are only going to become activated once they're cut, like in half, basically. That's the point of caspases, is that they're kind of folded so that they're not active all the time. And the big reason this is going to happen is T cell comes and kills you, or you've got a ton of damage in your cell and you should not pass on this cell another time. So this is a good little version, kind of discusses what we looked at here. So this is reception. This is the adaptation, or the adapter protein that's going to lead to cascade. Oh boy. This cascade is going to lead to more active genes like this caspase, and that's going to cause a physiological response, which in this case is apoptosis, and the cell goes down. So this is a good example of, although we're looking at physiological gene activating pathways, this is another physiology pathway or cell pathway that still has these same 1, 2, and 3 steps. 3 should be over here, for getting a small signal and triggering a very large response eventually. Okay, so you can look at these if you want. Come to me when you need clarification on these. So necrosis, remember, this is not an ordered process. This is just grievous injury. Something bad, really bad happened to the cell. All kinds of stuff is leaking out now. All these bad enzymes out of the cell are leaking. Everything's chaos. It starts hurting the cells around it. Apoptosis, remember, this is a pathway. We have the pathway and we can control it, usually. Again, in cancer cells, though, as we'll see, apoptosis is actually prevented so that they can keep surviving basically all this damage that they incur. And this is a good example, and this kind of ties into our cell cycle stuff, whereas there's a lot of on and off genes for the cell cycle to go. There's a lot of on and off genes fighting each other to make sure the cell either explodes or stays alive. So you can see like BCL2, for example, is going to stop the cell from dying, while the caspases, they're going to try and trigger the cell to die. So it's a big infight between genes and a big balance fight. And it's kind of the same thing between the cyclin and CDKs that say go, 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 make more cells, and P53 or other tumor suppressors or stop genes that say no, slow down, there's DNA damage, we need to kill the cell. So that's the big thing for today, or for this one. And we can see that resisting cell death is one of the key cancer hallmarks. And also, as we see in the next lecture, sustaining those proliferation pathways, that's another key. So when you put these two things together, that's when you're really, really in trouble. The other stuff is good, and like, we talked about metastasis, we talked about DNA immortality, we talked about how cancers need to eat, they need nutrients, so that's why you need to have blood veins showing up. But for the most part, if you have these two things, you're going to start making a tumor.